Tonicity, you need to understand the meanings of these words and then apply them to situations. So you already know hyper means more, and I'm sure you probably knew that hypo means less. Iso, in this case, is what we use to mean equal, and tonic refers to concentration, and by concentration, I mean concentration of solutes in a particular solution. So in other words, if you saw the term hypertonic, it would refer to something having or being more concentrated than something else. If something was hypotonic, it would be less concentrated. It has less solutes dissolved in it than something else. So that is the meaning of the terms hypertonic and hypotonic. If the concentrations are equal on both sides of a membrane, that's called an isotonic solution because the concentrations are equal. You would never be asked, you would never be given this, a cell, and that this cell has 10% solutes, and then they ask the question, is this cell hypertonic or hypotonic? That doesn't even make sense. That would be like me saying, am I taller or shorter than who? You have to be comparing it to something. So you could never be given this, but you could be given this, where you're given a cell, you're told that this cell was put into a solution, and that the solution, let's just say, contains 20% solutes. So now you have a cell with 10% solutes, you have a solution with 20% solutes, and then you could be asked, is this cell hypertonic or hypotonic to the solution? So does it have more solute or less? Less. So would it be hyper or hypo? Hypo. So this cell would be hypotonic. So this would be hypotonic. And if you were asked what the solution was, the solution would be hypertonic. So the cell would be hypotonic, the solution. If one is hypo, the other has to be hypo. All right, so the rule is that water will always go from the side with more water, because this is osmosis. Osmosis is the movement of water from higher to lower concentrations. Water will always go from the side with more water to the side with less water. The side with more water would have less solute. In other words, this would be the hypotonic side, the less concentrated side. And the side with less water and more solute is the hypertonic side. Because hyper means more and tonic means concentrated or more solute. In other words, water will always go to whichever side is hypertonic. Yes. So what's going to actually move is the water. You're usually going to be given a situation where they're going to tell you, if we go back to this drawing we had a moment ago, they're usually going to give you a situation. So let's say this is 10% solute, this is 20% solute. They're going to tell you that this is a cell and this is a semi-permeable membrane and that the solutes are unable to pass through the membrane. So then they're going to ask you, which way will the water go? That if only water can pass through the membrane and water can pass through freely, will the water go into the cell or out of the cell? The mistake people make is they look at the percents and they go, oh, well, water will go in because the percent is lower. But it's not the percent of water that's lower. It's the percent of solute that's lower. Technically, what's the percentage of water inside this cell? 90. So this is 90% water. And outside the cell? 80% water. So if water's always going to go to the side with less water, water should leave this cell. Now remember what I told you. We said that outside this cell was hypertonic, and inside the cell is hypo. So wouldn't it be true that water is going to go towards the hypertonic side? Water will always go to the hypertonic side. You will not have to. I mean, you can if you want to, but technically you're just wasting time if you go through and you start calculating the percents of water. Once you know which side is hypertonic, that's going to be the side with less water. Hypertonic means more solute. In other words, in comparison to the other side, that side has a higher concentration of solute, which means it's going to have less water. That's what the definitions mean. We're going to do a couple practice problems, but that's sort of an introduction to it. So, again, to clarify, if the solution is isotonic, that means that the concentration of solute is equal on both sides, which would mean that the concentration of water is also equal on both sides. And if you're asked which way water's going to go, the most correct answer is, even though there will be no net osmosis, water would be going in both directions at the same rate. So it's not that water has stopped moving, but there's not really going to be any change in concentration on either side because the amount of water going in is equal to the amount of water coming out. If it isn't isotonic, then that means one side is hyper and the other side is hypo. Again, it's not hard to figure out. All you have to do is figure out how much solute there is. Whichever side has the lower percentage or molarity of solute, that's going to be your hypotonic side. And that's going to be the side, by the way, since it has less solute, that's going to end up being the side with more water. Water will always tend to leave that side and overall go towards the hypertonic side, the side that has more solute and less water. This is true 100% of the time. All right, so these are the problems that you have in your notes. So you have a situation, these are supposed to be cells in beakers of liquid. So for starters, if we label these A, B, and C, which one, A, B, or C, is isotonic? Which would mean equal, correct. So A, thanks Carlos, A would be isotonic. If you were asked which way water would go, your answer would be that water would be traveling equally in both directions. So you would have no net change in concentration. All right, so now we go to B. So is the solution hyper or hypotonic to the cell in B? Hypo, exactly. So the solution is hypo. That would make our cell hyper. Which way should water go then? In or out of the cell? I hear both. The rule is water will always go towards the hypertonic side. So water will go into the cell. Remember, you can always double check yourself. Technically, what's the percentage of water outside this cell? 98% water. And inside, only 90. So it makes sense that water's going to go in because it's going from where there's more water to where there's less. But that is the hypertonic side. And then if we look at our last one, obviously this is going to be the opposite situation, right? Outside the cell is hyper, inside the cell is hypo, and so in this case, water would go out. So in B, the water would go into the cell, just to clarify. In C, the water would go out of the cell. So this is a sample of the type of problem you could see. Another kind of problem that's a very common one is they give you one of these YouTubes. They would always give you some kind of a little paragraph above it explaining, you know, a scientist did blah, 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 this is what was put in it, and they would give you information. They would tell you that a semi-permeable membrane separated the two sides. And in this one, they kind of threw you a curveball. Because rather than just saying such and such percent of salt or sugar or solute, they put two different solutes. And they used molarities. Don't let molarity throw you off. You don't have to be able to calculate molarity. If you failed that test in chemistry, you don't have to worry about it. All you have to know is, just like with percents, the higher the molarity, that just means it's more concentrated with solute. So the first question they ask is, which side is hypertonic at the start? They want to see if this is going to throw you off because they gave you two different solutes. All you've got to do to get the total concentration is add them together. So if we add together our left side, our total concentration is 0.9 molar. And on the right side, it's 1.2 molar. So based on that, which side, A or B, is hypertonic at the start of the experiment? B is hypertonic, exactly. Because there is a higher molarity on side B at the start of the experiment. And if only water can pass, we'll come back to question 2. Question 3 says, if only water can pass, which way will it go? So will water go towards A or towards B? Water will go towards B, because water will always go towards the hypertonic side. So once you determine which side's hypertonic, that's the way water's going to go. Again, technically you could treat this just like percents. You can subtract from 100. It doesn't quite work the same way, but you would see that there's less water on B than A. So water's going from where there's more to where there's less. But if you just remember that water always goes towards the hypertonic side, it would never be an issue. Now, question number 2 asks, if glucose can pass, which way will it go? If they give you one of those, where they give you a solute and they tell you that particular solute is able to go through the membrane, your rule is the same rule that applies to everything else for diffusion and osmosis. Anything that can pass is going to go from where it's higher to where it's lower. So based on that, here's our glucose. It's 0.4 on the left and it's 0.8 on the right. Will it go towards side A or towards side B? And the answer is it would go towards side A. So anything that can pass through the membrane, you're going to automatically assume it's going to go towards the side that has less, including water, but it's just that water is much harder to visualize because they're always giving you the concentrations of the solutes. They're not giving you the concentration of the water. So that's why you figure out the tonicity, and then you can get the answer from there. So anything that can pass through a membrane will go from where there's more of it to where there's less of it. That's just natural passive transport because of molecules moving. So water will go from where there's more water to less water, and any solute that can fit through the membrane will go from where there's more of that solute to where there's less of that solute. So that's the rule. So that is tonicity in a nutshell. We're going to do lab on this, like I said, tomorrow, where you're going to observe water and solutes passing through membranes. Tomorrow and Friday, both. The last part of this is actually about how cells deal with water. Let me pause this. So since water can freely pass in and out of cells, organisms have to do what's called osmoregulation, meaning they have to control their water balance. This is really, it's kind of memorization, but it's a little application too. Any organism that lives in fresh water, the problem they're going to face is that water is going to tend to want to come into their cells. So you may remember the paramecium. This was a protist. It had the central or the contractile vacuole that would swell up and squirt out excess water. So that's one example of a way that an organism would osmoregulate in fresh water. Freshwater fish have the same problem. They are living in fresh water, which is hypotonic to their cells, and they're constantly running that water across their gills. They're drinking that water. So basically what they do is they just make huge amounts of urine. They pee all the time. And that way, they can get rid of any excess water that they have in their system. What's going to be the problem for a saltwater organism?
Too much water in the body.
Right, that they're going to have water in the body that's going to tend to want to leave and go into the salt. So, what they do is they maintain high solute concentrations. Organisms that live in salt water tend to have higher concentrations of solutes in their blood. It's not necessarily high salt concentrations, but by having extra solute, it basically makes an isotonic environment so their cells don't shrivel up. They maintain high solute concentrations so that they're isotonic with the environment. The other thing that a lot of saltwater organisms can do is they actually have salt glands to excrete excess salt. Like saltwater crocodiles that are drinking salt water, they will literally make crocodile tears. The term crocodile tears for somebody like that's fake crying. It's because crocodiles can look like they're crying because they have these salt glands that excrete extra salt because they're drinking salt water. If you drink salt water, you won't have crocodile tears. You'll just be dead. But they have crocodile tears, and they can get rid of extra salt. Mangrove trees that grow in salty waters and other organisms like that have ways of getting rid of extra salt and ways to try to hold on to their water so that they don't get dehydrated. All right? And last but not least, there's a contractile vacuole. Plants, instead of exploding, like Nico said, they basically just create pressure on their cell walls.